his bachelor's degree. He worked for a while at Stanford University doing sequencing. Then he came to do a PhD here in Curtis Suttle's lab, where he's done more sequencing and analysis of sequences. And so he's been studying these really cool mineral formations in lakes that these formations happen as a result of biological activity. And he's been looking at viruses, I guess mainly phages, bacteriophages, associated with these microbialites in lakes in BC mainly, and also in Central America.
All over the world.
All over the world. So Rick White, take it away.
Thank you, Tom. I think I'm going to start off with just kind of an anecdotal thing. Is that if you had told, if I had met myself 5 years from now and would have said that I would be here in BC working on these things, I'd have called myself insane. But basically, this is how I'm going to start it out. And so basically, it's, as Tom said, a global survey of modern microbialites, form and function. And I work in the Subtle Lab. Of course, I won't be able to talk about viruses, which is what our group kind of focus on. But today, I won't be able to. So let's just kind of start them out. Click to work. So, of course, Earth, and it's important for our survival. And basically, we live amongst, as what Steve Hallam calls the sentinels of our world. And basically, if you look at these are the cellular fraction of the bacterial cells, and basically you have 10 to the 30 bacterial cells that's suggested on the planet. And among that, and where Curtis Suttle's work has come in to try to elucidate the immense amount of viruses in this kind of global biosphere. And that's 10 to the 31 viruses just in the ocean alone. It's probably orders of magnitude more if you account lakes and fresh water. But if you take anything from my talk today, learn this, and that we are on a microbial planet, is that the air we breathe, the water we drink, the food we eat, even our immune system is entirely able to be dependent on these organisms and our existence. So if you look at as far as geological time, and that's where I spend a good amount of my time thinking about, I'm going to give you a kind of rough overview of our biosphere as far as geological time is concerned. It starts at around 4 billion years ago with the end of the heavy bombardment. So this is basically the planet is starting to cool and things are starting to come together. Within an aqueous environment, we had origination of an RNA world. Basically, catalytic RNAs, they're basically starting to do enzymatic reactions. From there, the birth of DNA, and then membranes. From there, you have the first biochemistries, and at this point, we're talking about an entirely anaoxic environment, no oxygen in the environment. From there, there's some of the most ancient chemistries we know of, from stable isotope probing, is sulfate reduction, methanogenesis, and inoxygenic photosynthesis. At around 3.5 billion years ago, we have the first stromatolites. And what is a stromatolite? I'll get into that. It's a type of microbialite. But if you look at this sky, there'd be no oxygen in the air, there'd be acetylene. This would be a completely alien environment. If we even looked at Earth at this time, we would say this planet is uninhabitable. Basically, from that point, and if you read new paper from Nature out of Casey's group, we find that around 3 billion to around 2.8 billion years ago, we have the birth of anoxygenic photosynthesis. So the first cyanobacteria and the birth of what we call modern microbialites. So the stromatolites that we see in the fossil record in Western Australia basically occurred around 3.5, and so we have a billion-year gap in between here where there's no oxygen going on and no cyanobacteria going on in this picture. From there, from this increased activity from cyanobacteria, we had this thing called the Great Oxidation Event, which is the greatest mass extinction in the history of our planet. 99.9 with 1,000 nines of all the species that we know of today were gone at this event. At around 1.5 billion years ago, so we had the first photosynthetic oxygenic eukaryotes, so protists and algae and these kind of things, and eventually they conquered land at around 1.2 billion years ago. At 700 million years ago, as far as geologists are concerned, we see a complete decline of the dominance of stromatolites all over the world. But do we really? It's a question I want to figure out. But from all of this, for 85% of Earth's history, these structures have been present and representing the oldest continuous biogeochemical process in the world. And so basically taking CO2 and fixing it into these structures. So if you look at this, these are microbialites. So geologists have basically looked at, even as late as 1996, basically said that these formations are completely abiotically formed. No biological mediation involved at all. And we want to kind of put that on its tail and basically say that's not entirely true. And we want to have a modern definition of these things as far as microbialites is concerned as a sedimentary deposit secreted as a result of a benthic microbial community. So you have this extremely complex, ubiquitous microbial community that basically is in the right chemistry, precipitating these structures. And further, these structures are classified by what they look like. So stromatolites, it means stromas or layers. So you have these nice kind of layered projections. Dendrolites, so you have these kind of dendritic projections, and try to Google that and you'll get all kinds of weird things. Thrombolites, which are basically thrombus meaning clot, so things kind of coming together in a clot. And this is kind of most of the stuff I see in fresh water are thrombolytic structures. And then something in between stromatolites is this rare one called a mucosolite, which is basically a layered structure that basically forms these kind of nice circles. So if it's completely abiotic, then how is there evidence that it, going back even to 1973 with the Bouquet paper, showing that if you take just regular old soil and you isolate microbes and put them in this condition, you actually get calcium carbonate precipitation. They actually found 200 unique strains of bacteria just from soil that can do this process. But basically biologically mediated with the right chemistry, but entirely ubiquitous. So that leaves a problem for us, is trying to figure out how these things function. Here's a beautiful thrombolytic structure from Kelly Lake, and if you're wondering about how big this is, so we're talking around as big as this table is what we're talking here, as far as size is concerned. So these things are huge. Every ones that I've looked at, and so I'll give you kind of the current model of how we think this thing is going on, is from Duplass et al. In 2008. And basically, if you look at a freshwater ecosystem, you have this thrombolytic structure with a thick layer of cyanobacterial mat forming EPS, so extrapolymeric substrate, a carbonate core made mainly of calcium carbonate. We do see some weird things like aragonite, which is an isoform of calcium carbonate, as well as hydromagnesite, which is really weird. It's a hydrated magnesium compound. And then you have this kind of white snow. Basically, it looks like cement. If you picked it up in your hand, you think that you're making cement. From there, the abiotic factors, some critical stuff that you have to have. Of course, that falls out. In order for this to work, you basically have to have an alkaline water column, so basically higher than 8 pH. You have to have dissolved carbonate and dissolved either calcium or magnesium for the hydromagnesite. From looking in at this EPS layer, it goes from aerobic to anaerobic, so you go into the rock layer and it gets more anaerobic. In the case with most of the ones I look at, we don't see any real stratification of the water column, basically aerobic all the way down. And this is from Kelly and most of the ones I look at. From there, we have to have cyanobacteria. Cyanobacteria are basically providing the nutrients and fixing the carbon from these very allotropic environments. Also, forgot to mention, these environments are very allotropic, so very low phosphorus, and then basically the water column is a complete desert. So by having the cyanos kind of fix themselves on a hard surface, you amplify the water column such that you trap carbon from light, you trap light from photosynthesis, and then you trap carbon from visco in order to make that energy. As a byproduct of this metabolism, because the inorganics are there, you basically make calcium carbonate. And of course, cyanobacteria are the big one. They make the filamentous max and provide those nutrients, as well as protists that are kind of launched in and try to make a living as well. As a secondary event that also occurs in these very complex microbial communities, you also see anoxygenic photosynthesis. And this is actually the anoxygenic photosynthesis that synthesizes the alpha proteobacteria, for example, are the most dominant organisms in these ecosystems, as well as in the water column itself, and even in the ocean. SAR11, distant relative of these guys, basically dominate numerically all over the world. So basically you're taking H2S, which is basically that rotten egg smell, you're using light, and then you're capturing it through photosystems, and then fixing carbonate through rubisco. As a byproduct of this metabolism, again, you're making calcium carbonate, as well as you can have green antigenic phototrophs in the form of chloroflexes, which most likely are the remnants in these ancient stromatolites. Another thing is that you have these two ancient metabolisms that are kind of co-mingling with this more modern metabolism with photosynthesis, and that's sulfate reduction. So you trap sulfur through this kind of complex mechanism that you convert it with DSR into H2S, which then can feed an oxygenic photosynthesis and kind of into a feedback loop. But as a byproduct of this metabolism, you make calcium carbonate. And of course, the delta protease bacteria, so your disulfovibrio, your disulfomonas, these are the ones that are basically trapping this sulfate and actually making the structure. So any increase, any slight increase in the pH increases alkalinity, which increases precipitation. That's why this process, in general, is very ubiquitous, which makes it tough to kind of figure out, okay, who are the folks involved and how are they doing it? When I first looked into this, basically everyone kind of told me about it. You ever heard of stromatolites? I remember that, like it was just yesterday, I walked into Curtis's office, and he's like, well, I have this one project in northern BC. Have you ever heard of stromatolites? And I go to him and I'm like, absolutely not. No idea what you're talking about. And he said, well, what about the ones in Shark Bay? And that's these guys right here. So for here to kind of give you some ideas is that I basically mapped the topology of like the active structures that we see today that are actively making the structures. So we have in red freshwater, in green marine, hot springs in yellow, and then hypersaline sites in blue. It's kind of a misnomer because the ones in Shark Bay, where the oldest stromatolites are found, are also kind of hypersaline marine, so it's kind of in between. I spent a lot of my time in northern BC and as well in the Yukon. Sampling these guys. I also have some sites in the Antarctic, but I'll kind of course, going the wrong direction. Okay, there we go. So Shark Bay. So I have some collaborators in Shark Bay, and it's kind of got this neat little pronged A-looking structure, and because it has this upward angle, all this salt gets trapped in, but we get these kind of columnar stromatolites being formed. From there, we have two permanently frozen freshwater lakes in the Antarctic, Lake Vanda and Lake Joyce. Won't be able to talk about most of these sites today because I'm going to run out of time, but if you're interested, I can definitely talk about it. Algachica is kind of an outgroup, so we don't find the calcite in these structures. This is the only site where you make hard stromatolite and thrombolytic structures that are made from hydromagnesite, which is a magnesium compound. Very, very interesting Mexican system. Pascal Lake, which is very, very alien, hot, kind of not quite completely alkaline water column, but you get these coastal-like structures being formed. My outgroups from all of this are basically 3 northern BC lakes that at one time at least had the structures, but something changed in the chemistry such that the mats started dying and the structures themselves started to dissolve. So the chemistry is pretty much correct, but the microbial fraction may not be correct or vice versa, thus leading to the dissolution of the microbialytic structures. From there, from northern BC and Atlan BC, we have these kind of tri-layered mats, and they don't form full microbialytic structures. They kind of form in between because there's magnesium and calcium kind of coming in at the same time. But you get these beautiful layered, kind of soft yet calcified mats. As well as Clinton Creek, which is kind of a unique one, which is basically you have an old asbestos mine where they cut out this kind of trench, and then over 25 years you have these thrombolytic structures. So it's a man-made site by principle, basically. Rain fell in, and you got the structures. These are in 25 years. So normally these things take tens of thousands of years to form. Forgot to mention that. But in the case of Clinton Creek, it only took 25 years. Pretty interesting. Kelly Lake, which is in northern BC, another beautiful thrombolite, and actually the picture you saw before was from Kelly. Kelly Lake and then Pavilion Lake is where I spend the majority of my time thinking about these things to develop proxies for the entire survey. So when I first came in 2009, this was the only paper that was available, even looking at the function of freshwater or even marine stromatolites at this time. And basically it was one sample from Pesos Azules 2 and another sample from Reyes Marquesis. That was it, no replication. They did some quick 100 base pair sequencing, about 100,000 sequences, and that was it. And they hint at a lot of different things that are going on, but it was very incomplete. And without replication, it was hard to say. Plus, there's amplification, a lot of other things going on. But this is kind of, so basically started from basically nowhere when it came to the other data sets that are available. So methods, kind of going through, also forgot to mention that this thing we got involved with this because of a NASA CSA project called Mars Life. And basically what CSA and NASA wanted to do is basically use these rovers in Pavilion and Kelly Lake to kind of test out proxies to kind of go out and look for carbonate structures on other planets. And basically the thought is that on Mars, we see microbialite or carbonate structures. So if we can develop proxies on our planet to get at these structures, maybe on Mars we can say that there's biosignatures preserved on Mars. But that's about it. You have this beautiful rover kind of going in and looking at it. So what do? So I grab a microbialite from the pavilion or wherever I'm looking at. I grab 3 of them. I examine by microscopy, homogenize, extract nucleic acids. Initially tried to go after RNA, but that was kind of a no-go. Made all my libraries from hand, did 454 pirate sequencing initially, and then also kind of moved to Illumina due to the throughput and multiplexing. From there, we also wanted to look at the water around the microbialite as well. Because most people, they'll just grab a structure and they'll say, well, this is they'll do some 16S, and they'll say, well, these are the things that are there. But without looking at the sediment underneath and the water, you don't really know what is actually on the microbialite versus something that's just coming in there from the water column or from the sediment. And if the abiotic folks are correct, then they all should be the same, basically. Whether all the water or the microbialite shouldn't have any kind of unique structure to it. So once again, basically filter 100 liters of water, extract nucleic acid from the filters, and then library prep. And then we also do viruses. I won't have any time to talk about them today. Basically, anything that goes through the filter is basically viral. We use tangential flow filtration, which is a kind of weaving in and out of the structures through pore size, and you basically remove water. And from removing the water, you concentrate the viruses. From there, extract nucleic acids, and then make libraries. Another thing I won't be able to talk to about all the isolates that I also work on, where I actually get full genomes from isolates. I'll be mainly talking about the metagenomes, but I spend probably 99% of my time here in the dry level stuff. So from there, I basically be doing a hybrid approach. So I'm taking all the different Illumina technologies, because you can take one library and sequence them on all the different platforms, and also 4x4 titanium reads, and basically removing all the crap. So Phi-X, E. coli, anything that's the spike in control, I have to remove, and then any poor quality reads. From there, I assemble with Ray. I map the reads back to the assembly, and then for my genomes, I align to a reference if I have it. If I don't have a reference, I'm basically doing it completely de novo. From analysis standpoint, when I talk about database, I'm talking about a blast base. So I'm taking a known database like KEGG or Metaseq or one of these other ones, blasting it, or a better form is LAST, which is way faster. And then Steve Hallen wanted me to look at a non-database approach. So from there, basically looking at GC content or protein isoelectric point prediction to kind of get an idea of how to separate these very complex microbial communities. Learn R now. If you don't know it, it's fantastic for figures. And then STAMP is another program list. So with all science, you're supposed to start with a question, which is kind of good and new. So I'm going to start out with this one. Is there a unique microbial community specific to the morphology of a microbialite? And you're like, okay, what do you mean by that? So in Pavilion and Kelly Lake, we have these unique morphologies of the structures. So basically, as you go down the water column, you start from this kind of lower lettuce bed kind of thrombus structure. From there, you get this kind of round, kind of cabbage head-looking thing. And then you get asparagus kind of leaflets at 25 and 20 meter depth. And then at 45 and 25 in Pavilion and Kelly, of course, Kelly is not as deep as Pavilion, as you can tell. So you get these kind of artichoke leaflet-looking structures. So the first question is that if you have unique microbial community associated with the structures that you see as a function of depth? That's the first question. And so you look at this, and it's R and it's ggplot, and you're like, I can't tell the difference at all either. So I did this. Kind of maybe look at some subtle differences, and really not much kind of going out and saying, wow, I'm different. Maybe a little bit more cyanobacteria at this depth, but really very, very similar as you go across the depths. Okay, So basically, taking the metagenomic reads from the proteins, predicting orbs, and then trying to blast them against a known database. So similar, basically similar as you go across. So I did this kind of nice little t-test analysis with STAMP, and basically wanted to look at the differences between Billion and Kelly. So on a class level, basically very, very similar, about 98.98% R score, and basically only a few things that are different kind of on this stretch. If you look at a class level distinction, but cyanos kind of pop out, and as you saw before, they kind of popped out again. And you basically see that kind of some of the ones that are involved in the chemistry, such as maybe alphas or betas or deltas, are kind of popping out. When you go to function, they're basically very, very similar as far as function between the two lakes and the two ecosystems. If you take all the replicates, all 3 replicates from each depth, and you compare them on this ordination plot. Two kind of things that kind of popped out were kind of interesting were these heavy metal resistant genes, a serine gluconate cycle, and then tRNA modified protein. But these are kind of one-off in Pavilion. And of course, in orange, Pavilion, and then blue, Kelly. I've been working with the Howland Lab a lot, and they have this new thing called metapathways. It's awesome for looking at metagenomes. And basically what we did here is we basically compared all 3 replicates from all the depths and basically did a nice little Venn diagram for the metaseq pathways, and basically found that most of the main pathways that are found in both ecosystems are basically shared. And there's very few pathways that are unique per depth, which is kind of interesting. So once again, it's a very ubiquitous process, so it's kind of hard to say, okay, here's the smoking gun, here's what's going on, but we know that right off the bat that a lot of these pathways are shared between all the different structures. So then I kind of wanted to narrow in and really try to go after this question. So I looked at these artichoke morphologies, and I looked at everybody else and compared them crosswise, t-tests, all these really fancy statistical tests. Basically didn't find much until I started looking at the artichoke morphology. What I found right off the bat is that I can go all the way down to the genus level for the artichoke morphology, and I get a very, very high correlation between the two, and very, very high functional relationship in the KEGG database. So once again, some things that pop out, Microbialites, Nitrospira, Orchidaria, but if I do the same thing with any other structure, I don't get this high correlation point. So could be very interesting there. With KEGG, once again, some interesting things popping out that are different from the other two component systems, APC transport and oxidative phosphorylation. So it appears that, at least with the artichoke morphology, that there could be something. They're more similar to each other between the 2 lakes than they are with other depths within the same lake. Okay, it's kind of confusing maybe. Basically, they're more similar to each other than if the depth above them, which was kind of interesting. So could be something there. Going in and trying to say, what does the metabolic potential of the community differ between the surrounding environment? So going back to that water versus sediment question. So going back to what Steve Howland kind of told me to go after is try to look at and try to separate metagenomes, and you can kind of separate them by GC content. So I did a quick little GG plot here of looking at the GC content between the filter or the water column, the water surrounding the microbialite itself, and then the sediments. You have at this 10, 20, 30, 20, 25, and 45 GC content. So if you look at the the predicted protein isoelectric points, very, very similar across the depths. But if you look at GC, we see we can easily separate them by what they are based on GC content. Back to the stamp plot. So this is a NOVA with a Turkey-Elmer and an ED2 squared, and with a p-value of 0.05. And basically, we take the metagenomes and we basically take all 3 replicates and surmount them into one point on the plot. We have our microbialites in these kind of green triangles, the sediments and the filters, and we basically see that with this kind of PCA ordination, that the microbialites from Pavilion Lake kind of cluster very, very close to each other, away from their surrounding sediments and their water columns. So the microbialites themselves, the microbial community on the microbialites, looking at the structure of the microbial community, is very, very different than the sediment and the water column. We can also look at it on a functional level using the seed classification, and once again we see the same thing. Microbialites cluster together very, very tightly, whereas sediments are kind of out there all over the place, and then the water column samples are more close to each other than the microbialite. So